Hi everyone, I am Huwaida Hanifah, S50685, would like to present my final year project entitled Preliminary Data on the Diatom's Response to the Introduction of Cadaveric Derived Materials Associated with the Decomposition of Partially Submerged Buried Cadaver. I am supervised by Dr. Siti Sofu Ismail and co-supervised by Dr. Nurul Shahida Rizwan. So I'm going to start with the introduction. The buried body will undergo 5 stages of decomposition process which help in organizing the postmortem changes and any information related. The decomposition process of 5 stages are initial decay, putrefaction, black putrefaction, butyric fermentation, and the last one is dry decay. These 5 stages describe the response of diatoms to the introduction of cadaveric derived materials with decomposition of partially submerged cadaver. The problem statements are the study of partially submerged cadaver buried in a mangrove area is lack of research compared to those in terrestrial area and drowning water cases. The inconsistent variables may alter the decomposition process due to the season change, and this might lead to difficulties in estimating the postmortem interval. My specific objective in this study are to determine the diatom species associated with the decomposition of a partially submerged buried cadaver in a mangrove soil system and to elucidate the response of diatoms to the introduction of cadaveric derived lipids at each decomposition stage. Generally, the study involved 5 steps in methodology. The study was done at mangrove area of Biowalk in University Malaysia Terengganu. Both pictures indicate the Biowalk map and also the burial site in mangrove area. For a simulated burial experiment, 20 grams of pig fatty flesh was buried in pots, partially submerged in a mangrove soil system at depth of 15 cm from the surface. Each burial was replicated 3 times along with the control soil. I continue with the next slide, which is the steps in diatom extraction, where the topsoil collected was added with formalin, centrifuged with many steps ahead such as addition of distilled water, potassium permanganate, and also concentrated hydrochloric acid. Prior to the centrifugation process, the samples were placed in water bath for 24 hours. Then, the diatom samples were obtained. Next is the method for lipids extraction by using black dye solution. Turning to results and discussion, the graph of post-experimental soil pH shows the different initial pH value between the burial and control soil. The increase of pH value was due to the ammonium release during the ammonification process, while the decrease may be due to continual degradation of tissues. For the total lipid extracts, day 20 remarks sharp increase in TLE concentration corresponding to the putrefaction stage. This may be due to microbial community's response. The later stages of decomposition process results in low TLE concentration, less amount of the introduced cadaveric derived materials, and also the formation of adiposauri were observed, and this was due to the lesion in the periflesh. The decomposition rate shows increasing trend in between day 1 to day 5, corresponding to initial decay stage. This was due to the maximum degradation activity of microorganisms happened, which result in a greater mass loss by the periflesh. The declining trend of decomposition rate within day 12 and 120 with minor fluctuations could be attributed to the cadaver's depletion of nutrients, which reduced microbial activity. This graph of diatom diversity versus decomposition stage shows the decrease in number of species from putrefaction to the black putrefaction stage, where the less microbial activity happened. The rich nutrients released from the pig periflesh and less surface area free for colonization. This pie chart indicates the dominant species occupied in control soil, which is Gyrostigma species. Next is the dominant species occupied in burial soil, which is Coscinodiscus species. This graph proves that both control and burial soils indicate the existence of diatom species, regardless the burial of pig periflesh, considering the diatom's response towards biotic and also abiotic factors in environment. The diatoms played its role sensitively towards the environmental change. As for the conclusion, pH value for both control and burial soil became acidic approaching the end of burial interval. Rate of decomposition experienced rapid process at the beginning and became slower towards the end of 120 days of burial period. Dominant species found in both control and burial soil are Gyrosigma species and Coscinodiscus species, respectively. Progress in TLE concentration at early 4 stages of decomposition process related to the quick decomposition of cadavers, and the last one is diversity index of burial soil is higher than that in control soil. These are my reference. Thank you for listening. Have a nice day and stay safe.